Welcome to the inaugural talk of the seminar series on COVID-19 that we have put together at UCSB. The goal of the series is to examine the virus and the disease from a multidisciplinary perspective. We bring together a group of experts who will examine different aspects of the disease, such as medical, biological, modeling, social, over the coming weeks. We'd like to thank the UCSB administration and the Novim group for their help. The Chancellor will be saying a few words later today. Finally, I would like to remind you that this series has been put together in a hurry. It is not going to be perfect. Please pardon our mistakes. Over to Joe Walter for more details of the series and introductions of the speakers today. Thank you, George. Thank you, Joe.
Thank you very much, Ambujan. Thank you, everybody, for attending today. I wanted to share a little bit more about where you can get additional information about the series and some updates about things coming up in the future. Hope you can now see a page on the World Wide Web. The address should be showing pretty clearly on the top, citsucsb.edu/spring 2020. And we will be updating this page as we finish identifying the various topics and the speakers who will be joining us for this series. I'd like to enlarge a piece of that page just a little bit for your additional attention. And that gives you about in the middle of the page, there's a place where you can click and that link will take you to a site where you can sign up for our mailing list. That mailing list will be sending out weekly information, and we will also to be able to send out information when we are able to start sharing the recordings of these sessions as well. So please sign up and we'll be glad to send out information about this series as it becomes available. Well, that said, I'd like to say just a few brief words about our two speakers who will be addressing us today. The first will be Dr. Carolina Arias-Gonzalez. Carolina is an assistant professor in our Department of Molecular, Cellular, and Developmental Biology here at UC Santa Barbara. And Dr. Arias has a great deal of expertise in having studied virus-host interactions. And this is really a critical topic. We couldn't find a better expert to explain some of these dynamics to us than Dr. Arias. Dr. Arias will be speaking first, and then in the second half, following the Chancellor's remarks, we'll be hearing from Dr. Lynn Fitzgibbons. Lynn comes to us from Cottage Health here in Santa Barbara, where she oversees the HIV and infectious disease clinic here for the Santa Barbara County Public Health Department. She is an expert in infectious diseases as well as internal medicine and chair of the Division of Infectious Disease at Cottage Health. She will be sharing all kinds of insights with us today about approaches to the disease, how things are being remediated here in the county. Without any further ado, I would like to make the brief comments over and now turn it over to Dr. Arias. Thank you.
An introduction and an update to SARS-CoV-2 from, as Joe mentioned, the virological and the clinical perspective. So first, thank you to Joe, Ambush, and Chancellor for organizing this series. I think it's critical that we stay informed at this moment. And again, we'll do our best to share the most up-to-date information with you. And if you have any questions that we cannot answer today, please feel free to email me or us as needed. Okay. So I'm going to start by telling you and sharing with you some details about SARS-CoV-2, the virus itself. We're going to talk about what are coronaviruses, the origin of SARS-CoV-2, what we know about that, and some details on the diagnostic methods that we have available to test for SARS-CoV-2 and COVID-19. So let's just start with the coronaviruses. So we are constantly hearing about coronavirus everywhere, and they are not just one, but several. They are a very large family of viruses that infect both humans and animals. They get their name from the distribution of this spike protein right here that surrounds the shell, the lipid bilayer that protects the viral genome. So this is basically the structure or rendering of the structure of the virus. The spike protein right here that is arranged in a way that resembles a corona or a crown, and that's where the name of this family comes from. They have a single-stranded RNA genome that is of about 30,000 base pairs that encodes for about 27 proteins. Just to put it in perspective, your genome is a double-stranded DNA molecule of about 30 billion base pairs that encodes more than 20,000 proteins. So although these genomes are small and they encode for few proteins, viruses can cause havoc in the cells. Viruses or coronaviruses, as every other virus, are obligate intracellular parasites. And what that means is that they cannot replicate outside of a host cell. So here we have a rendering of the viral cycle, and this brown square represents a cell. You're going to hear from Lynn later on that these viruses are able to survive in surfaces and in the environment, but they cannot replicate unless they are inside of a host cell. For the SARS-CoV-2, we know that the process of infection starts when the spike protein recognizes the receptor in the cell surface. In this case, the receptor is the angiotensin converting enzyme 2 or ACE2. Once the spike protein interacts with the receptor, the virus is able to gain access to the cell. It releases its viral genome and it is translated. So we know that as I mentioned, the viral genome is an RNA molecule that can be recognized by the cell ribosomes and can be directly translated into the proteins that are needed to make more particles. Once this viral genome is replicated massively, so there are a lot of copies that are made, they are packed in here in this process that you can see. They are packed into new viral particles that are then going to be released from the cell, and the infection is going to be perpetuated. So the more we understand about this viral cycle, the better we're going to be prepared to repurpose or design new tools that are going to allow us to control the replication of this virus. Lynn is going to tell you more about some of these efforts later on, but I can tell you that, for example, we have heard in the news that now they are collecting the plasma from convalescent patients. That plasma contains antibodies that are going to recognize the structures in this virus and are going to block the interaction of the spike protein with the receptor. So we can block the recognition and the first steps of entry. You may have also heard about hydroxychloroquine, and this is a drug that is not an antiviral, it's an antimalarial drug, but there is some evidence out there that suggests that this compound is blocking the subsequent steps of entry. So once the virus recognizes the receptor, if there is a chloroquine or if its cells are treated with chloroquine, entry is not going to happen. We can also directly block replication, and this takes advantage of the unique properties of the polymerase of the virus that are very different from those that you're going to find in cells. So with some compounds that, again, are in clinical trials now, we can block this step of replication and obliterate viral replication completely. So there's a lot of research now going on trying to understand each one of these steps in order to control it. So now let's talk about the human coronaviruses. So as I mentioned, the coronaviruses are a large family of viruses. We have hundreds, if not thousands of viruses that circulate in animals. And we have 7 viruses that circulate or can infect humans. They can be divided into 2 groups. One of those is the common types. There are 4 viruses in here that are associated with mild respiratory disease and are the culprit for up to 30% of the common cold cases. The 229E and OC43 have been circulating among humans for a long time, and NL63 and HKU1 were discovered early in the 2000s. We also have a group of viruses, of coronaviruses, that cause severe disease. These 3 are the severe acquired respiratory syndrome, or SARS, the Middle East respiratory syndrome, or MERS, and the SARS-CoV-2, the causative agent of the COVID-19 pandemic. And we're going to talk about the origin of these viruses in just a moment. But let's focus now on the origins, or where do we think that the SARS-CoV-2 came from. So here I have a timeline, a short timeline of the first events in the initial outbreak in China. So we know that there were a cluster of pneumonia cases that were reported between mid to late December of 2019 in the province of Wuhan in China. Many of these cases were related to, or there was a cluster of cases that was tracked to the Huanin seafood market, and we're going to talk about in the next slide. This small outbreak of a few cases rapidly progressed, and by mid-January, we had hundreds of cases in Wuhan, and in January 21st, we had the first case reported in the United States. And as we know, the pandemic quickly expanded, and we are over 1.5 million infections across the world as of today. But I want us to focus right now here on the first cases that were identified in the seafood market. So this is the panel market, and in there are a lot of animals, wild and farm animals, that are sold for human consumption. So when there was the first report that there was this cluster of cases in here, people started quickly investigating if there was some animals that were infected that humans could have been exposed to them and acquired the virus. And the reason why people started looking for these animals is because of what we know from other epidemics of coronaviruses, in particular that of SARS and MERS. So SARS was first identified in 2002, and the origins of this virus were able to be tracked back to bats. So we know that there is a virus that is very similar to the human SARS that circulated in bats. It infected civet cats, and these civet cats were being sold in markets for human consumption. And it's thought that in one of these cases, the virus was able to jump from these civet cats to humans, and then the virus was transmitted in the population from human to human. Something similar happened in 2012 with MERS. We know that there is a virus again that circulates in bats that is very similar to MERS. What is thought that happened is that these bats infected dromedary camels, and then humans came in contact with these dromedary camels and acquired the virus. After humans acquired the virus, there was sustained transmission in the community. So what we think is that something like this could have happened with SARS-CoV-2. And I'm going to show you some evidence that supports the idea that this virus comes from bats. But how do we know that these viruses come from one animal or the other? And we do that, or we know that, by comparing the sequence of viral genomes from different isolates. And we can infer the relationships between these viruses. Good, thank you. We can infer the relationships between these viruses. So in the same way that I could look at samples of human DNA, for example, from you and your siblings and your cousins and your friends, I could infer the relationships. I could see who is closely related to you genetically, who is related to you but is not a direct sibling, and who is not related. So the same process that we use for this kind of genetic linkage in humans, we can use for viruses. So we have here a diagram of the viral genome and some regions that people have looked and compared between human viruses, bat viruses, and what is suspected to be an intermediate host, pangolins, in terms of the sequence conservation. So we are in particular, or researchers are in particularly looking at the sequence of the spike protein, that one that interacts with the receptor in the cell surface. And the reason is because this protein is highly variable. It mutates very quickly. So by looking at the changes that have occurred in this protein at the genetic level, we can track relationships between the viruses. And what we have seen is that the human SARS-CoV-2 virus is closely related to a virus that circulates in bats, here named as RATG13. These sequences, the sequences of the spike for the SARS-CoV-2, are also closely related to sequences found in viruses in pangolin and to those that have circulated in human populations before this SARS virus. This conservation is true not only for this small region here, but also for highly variable regions that we can see right here. So from this analysis, we can, we have evidence that this SARS-CoV-2 is closely related to viruses that are currently circulating in bats. So this is a scenario of what could have happened. So we know that very likely, with a certain degree of confidence, we can say that the SARS-CoV-2 virus likely originated in bats. It was then transmitted to an intermediate host that could be pangolins or other animals. Now we know, for example, that some cats can be infected as well as ferrets. So there could be other intermediate hosts. And that this was then transmitted to humans. Once the virus entered the human population, it is transmitted from person to person. There's a sustained human transmission. Another possibility is that there was direct contact with bats and that the humans acquired the virus from this population. And then again, there was sustained transmission. There are two possibilities as well on why this virus evolved or where the virus evolved to be transmitted among the population. One is the virus could have changed in animals. It could have acquired some properties that facilitated the infection of humans and the transmission among humans. Or it's possible that this virus was acquired by certain members of the population. It did then change in humans as it was being transmitted, and it became a better pathogen, let's say. It gained the ability of transmit very efficiently between humans. Either one of these possibilities is viable, and there's a lot of research that is being done right now to try to really dissect where this virus came from and if there are any intermediate hosts or not. One thing that is for sure is that the virus does mutate when it's in the human population. So this virus mutates very rapidly, and we can track the evolution and the mutations of these viruses by looking at the sequence of the viral genome. And this is what we have here. So these are about 2,000 viral isolates that have been sequenced, and the sequences have been analyzed to find relationships between these isolates. Here we have in the x-axis, we have time, and in colors, we have the different geographic locations where these viruses were isolated. So you can see that depending on where they were isolated, they are closely related to each other. So not only this tells us or shows us how the virus is changing and evolving, but it also gives us an idea of how it's being transmitted. And that you can see from this map. So again, in colors, we have the different locations where the virus have been isolated and characterized. And if you look here, for example, at the United States, we know that the isolates that we have circulated came from Europe or from Asia or Australia. So by just looking at the sequence, we can infer, again, relationships between viruses, and we can detect how it is being transmitted at a global level. Now I want to share with you a couple of slides on how we are diagnosing SARS-CoV-2 and COVID-19. And we're mainly using two methods. In one, we aim to detect viral sequences in samples, and this is going to allow us to monitor active infections, and we use a method known as qRT-PCR. And I'm going to tell you the details of that method in a moment. We also can detect antibodies in samples, and this is going to allow us to monitor exposure to the virus. We're going to be able to see what person was exposed to the virus at any given moment, and we do this using serological tests. So let's talk first about the detection of sequences, of viral sequences in samples. So we use the qRT-PCR, or the real-time reverse transcriptase polymerase chain reaction, which is a mouthful. In this method, what we do is we collect the samples, here a nasopharyngeal swab, and then we extract all the RNA that is in that sample. If you remember, I mentioned that the viral genome is an RNA molecule, and if you are infected, we're going to be able to detect those viral sequences or those viral RNA sequences in the sample. It is very hard to detect these directly, so what we have to do is convert these into DNA by a method called reverse transcription. And once you have DNA, here illustrated as a double-stranded molecule, we can amplify it using DNA polymerases. In this particular assay, what we use is a probe that is labeled with a fluorescent dye that is complementary to this DNA that we're looking for. So these will be complementary to viral sequences, to specific viral sequences that we have, that we know. Once the polymerase starts copying these sequences here, at one point it's going to hit this probe, and this dye is going to be released. Once this dye is released, it's going to emit a fluorescent signal. So in essence, what we have is, if you have these viral RNA sequences that we want to detect, they are going to be amplified. And the more sequences there are, the more fluorescence you're going to be detecting. And that's what is illustrated here. So for a positive sample, we're going to have a fluorescent signal that is going to come with time. We're going to be able to detect it using specialized equipment. But if you're negative, if you're not infected, these viral RNAs are not there, and you're not going to be able to detect any signals. This is a very sensitive assay. Is, as I mentioned, used widespread for the diagnosis of SARS-CoV-2, but it does have some limitations. The first one is that it requires a specialized equipment and trained personnel, so it cannot be run in any place, not in a doctor's office or in the field. And the second one is that if the viral load is too low, if we don't have enough molecules of RNA here, we might not be able to detect it. We might not be able to detect the virus. And this is particularly important in events that are pretty early infections, or in convalescent patients when the infection is already being resolved. So in cases like that, what we can use instead of the RT-qPCR or the qRT-PCR is serological tests. In this case, what we're going to detect is antibodies against the SARS-CoV-2 virus. For this, as we know, every time that you're infected, your immune system is going to recognize that there is a virus, so that you have an active infection, and it's going to respond. And one of those responses is the generation of antibodies that are going to recognize specific structures and sequences in the virus. In this case, we have some antibodies that are recognizing the spike protein. We can then look for these antibodies that is circulating in your plasma or in the plasma of infected patients. And that is done by using this kind of test. So this is a serological test that works in a similar way to something that would be kind of, or a pregnancy test. It works in a similar way like that. So what you have here is a reservoir for a buffer that is going to allow you to carry the reaction. And you have a little reservoir for a blood sample. So basically you just add blood here, and if there are circulating antibodies in the patient, they're going to interact with specific antigens, so little pieces of the viral proteins that we know are recognized by antibodies. If the antibody recognizes those elements from the virus, there is going to be a colorimetric reaction that you can read here as a band. So if this band appears here, there were antibodies in that sample, and the patient was somehow or exposed at some point to the virus. So these are the possible outcomes. So you have the presence of the test line. So there was an antibody circulating. You have a negative test. You have a control letting you know that the test did work, but you do not have any antibodies that react to the virus, or the test is invalid. What is really important is to remember that this assay reports exposure to the virus and not necessarily the presence of the virus in the sample. So it can be used for patients that are not actively infected, but that were infected at some point. It is really important to remember that widespread testing is essential, especially for the cases where we are going to need to update policies, like, for example, in the case of social distancing. So if we know how the virus is transmitted in our populations, and we know how many people have been exposed, we can make educated decisions regarding how to control the transmission and how to either strengthen or ease some of the prevention measures that we're carrying. And so with that, I want to transfer to Lynn. Let me see how we do this. Okay, so that she's going to tell you more about the clinical aspects of the disease. Okay.
So, so Lynn, before we get to you, I think the Chancellor's system should be working now. So let's see if we can have a few words from Henry.
Okay, can you hear me?
Yes.
Oh, great. You can hear me. Okay, good afternoon. Sorry, I think you must be able to.
Yes, we can hear you. Yes.
But you cannot see. Can you see me?
Yes, we can.
You can. Okay.
Right,
perfect.
right at the top of your head. So you may want to remove the camera a bit towards your face.
Okay. Is this okay now?
No, it's not. You're too high up. Yeah, now you're okay. Yeah.
Oh,
thank you.
Okay. All right. I'm just very pleased to join you for today's webinar. And this is the first of several to be held throughout the spring quarter that will focus on the issues, approaches, and the consequences of the COVID-19 crisis. We so much look forward to today's discussion. And thank you, Carolina. It was a wonderful talk you just gave. As we find ourselves in this unexpected position of navigating a global pandemic together, I hope you and your families stay healthy and well. I would like to thank all our collaborators of this seminar series who are working hard to keep us all well informed. This includes our own UC Santa Barbara faculty. Of course, I would like to thank Ambush, thank Professor Singh for your leadership and your efforts and sacrifices and your efforts to coordinate all of our faculty together. That's quite a bit of work. And also I want to thank Cottage Health colleagues. And I also want to thank our Novin team, especially special thanks to Jim Knight and Michael Didmore. And they are the one who contacted me first, and I connected with Professor Singh, then connected with our faculty members. And we have had several meetings already to come up with this idea to give this seminar. A situation of this magnitude requires an interdisciplinary approach, drawing on the ingenuity of artificial intelligence, machine learning, data science, and other fields, and big data and other fields to tackle various aspects of COVID-19. UC Santa Barbara is uniquely placed for this kind of a comprehensive collaboration with our so many brilliant minds throughout the community in each of these areas, and a campus-wide commitment to building up these interdisciplinary fields of study. As an example, toward the end of this year, we launched our Center for Responsible Machine Learning, and we also received a generous anonymous gift of $500,000 to support our data science and data analytic initiatives on campus. So we now find ourselves in desperate need of such innovative research at the frontiers of science in order to save lives around the world. And thank you all for your tremendous effort to protect our global community. There's no doubt that the world you are doing is in the interest, the work you're doing in the best interest of all of humanity. So I will now turn the podium back to Ambush.
Thank you, Henry. Those were very kind words, and that is the help that we need in terms of our efforts to solve this pandemic. And now it's over to Lynn, who is all ready to tell us about the second piece of the puzzle. Thank you, Lynn.
Thank you.
Thank you.
Thank you all. So as was mentioned earlier, I'm an infectious disease physician, and I have the tough task of following Carolina and her really fantastic description of the virologic aspects of SARS-CoV-2. When we designed this talk, we decided that she would speak obviously about the virus, and I was going to talk about the disease. And I've been speaking a lot about this disease, as you may imagine, recently. And as I prepared for this particular seminar, I think I increasingly became aware of the term this bad disease. And I want to take you on a tour through aspects of this disease that I think really bring to light just how strikingly different this disease is from any other that we have dealt with in really any of our lifetimes. So with that being said, I've been asking colleagues really since the inception of this seminar series, what renders an infectious disease a bad one? And it's interesting, you speak to physicians, to healthcare providers, to scientists, to lay people. People have very differing opinions on this. There are a variety of answers that arise. The first is simply the idea of a disease that causes lots of symptoms and suffering, severe symptoms and suffering. Influenza, for example, for anyone who's had influenza, severe headache, horrible muscle aches, high fevers for days and days and days, horrendous cough. Clearly this, by all measures, would be a bad infectious disease. Or is it more important that an illness goes on for a long time? Is it that a short-lived illness is not that bad? And if it causes these symptoms for longer periods of time, that it would be a bad disease. Hepatologists, for example, would argue that hepatitis C, while initially may not cause much in the way of symptoms, continues to cause problems in the liver, fibrosis over decades, and surely this is a bad infectious disease. Or is it, as we experienced during the horrors of the West African Ebola epidemic, that we had an infection for which we had no effective treatment or cure? Is that really what defines a serious bad infectious disease? Or as in the case of, for example, polio or meningitis in children in sub-Saharan Africa? Is a bad infectious disease one that, regardless of what happens during the period of symptomatology, leaves a person with very severe sequela or indeed permanent physical or neurologic disability? And then shifting focus. Is it actually very little to do with the clinical aspects of the disease, and is it more to do with how infectious or transmissible a disease is that renders it bad? Is it that a very contagious virus, such as the measles virus, is that really what makes a disease bad? Is it that it's silently contagious? During the horrors of the HIV epidemic, we had patients who would become infected with HIV and then go on to live for years or longer, sometimes a decade, with very little in the way of symptoms, while they could themselves, unfortunately, go on to infect others. Or is it simply that a bad infectious disease is one where we have the unknown, where we have poorly understood transmission routes? And then finally, from the epidemiologic perspective, is it that a bad infectious disease is a disease that impacts vulnerable populations? Is cholera a bad infectious disease because it ravages communities in the developing nation, kills hundreds, thousands of children? Is that what defines really a bad infectious disease? Or is it simple? Is it just case fatality rates? Is it that a bad infectious disease is a disease that kills people? And finally, I think the question on the table today, is it actually the extent of a population level infection or pandemic spread that really, above all else, tells us this infectious disease is the bad and perhaps the baddest of the bad? So let me ask you to consider COVID-19. And COVID-19, I think everyone's familiar with the terminology, but COVID-19 is, of course, the name we have given to the disease that is caused by the SARS-CoV virus, the COVID-2 virus that Carolina described. So let's take symptoms and suffering. What are the symptoms and suffering? What is the disease that we are seeing on the clinical side when patients are coming in here or around the world with this disease? And here we have a nice compilation of the data from about 55,000 lab-confirmed cases in China, where 88% had a fever, 68% had a dry cough, 38% fatigue, about a third sputum production, and I'll let you read the rest, but of course, we've heard this. We have shortness of breath, we have muscle aches, sore throats, headaches, chills, nausea, vomiting, nasal congestion, diarrhea. Several subsequent case series have described other symptoms, including more in the way of diarrhea or GI symptoms, perhaps anosmia, where patients lose their sense of smell or of taste. And so these really, I think, are the primary symptoms of this disease. The Chinese researchers and clinicians have been phenomenal in very quickly getting data to those of us around the world who are seeing patients with this. The Italians have done the same. Many countries have really pushed hard, not just to care for the patients suffering with this disease, but also to share this information as quickly as possible. And this publication in the New England Journal of Medicine, now about 5-6 weeks ago, was really the first big study that looked at the clinical characteristics of those with disease. This looked at about 1,100 confirmed cases from, these were all hospitalized patients from around China in 30 different provinces. 47 years was the average age, and we continue to see this. We see a slight lean towards more male prevalence and more male incidence than females, as they saw. I'll let you read the common symptoms, again, consistent with the other data. And the median incubation period, about 4 days. We also know what is happening with critically ill patients. I've described thus far big populations of patients with lots of symptoms, hospitalized patients, but what about the sickest of the sick? And this was published in the Lancet Respiratory Medicine. This was a group of patients, and I'm not going to take you through every piece of data and every one of these slides, but the slides can be made available on the website for your review as well as for your reference checks afterwards, if anything is of particular interest. But ultimately, this group landed on 52 critically ill patients who had SARS-CoV-2. And here I just show you that they broke this down in the left column compared those who survived to those who died versus the total. And you know, as we're seeing in the CDC data, there was a lot of chronic medical illness, more so as we would expect in those who died. You also see the age breakdown, and we're going to talk much more about that. This is a second table from this publication. Again, anyone with interest, please do read the article. I thought it was very helpful. Much more of an observation than any sort of deliberate controlled study, but I think very, very helpful nonetheless. I think what struck many of us who were facing this epidemic in our own communities was really this very, very dismal look at patients who were mechanically ventilated, and how few of those, meaning their lungs stopped working by themselves and they had to be put on a breathing machine. And again, how poorly this cohort of patients did once they needed a ventilator. And then I'm going to just point out one thing from this third table, which is the duration of symptoms. So here, or the duration of ICU, excuse me, presentation. Very long stays in the ICU, 9 to 11 days on average. And then what about the CT scans of the chest? There's been, again, a lot of early publication on this. And here we see a CT scan, which I think many are familiar with, but many may not be. A CT scan is essentially going to take a slice of a patient and give us granular detail with good resolution of what's going on really inside the patient's thorax here, inside their chest. And so this study looked at 81 patients, and they basically trended what the CT scans did over the patient's course. And they showed that early in disease, it was more likely to affect only one lung, and really to begin with what we call ground glass opacities. And here I point out these really subtle, slightly gray areas where most of the black lung here would be regarded as healthy, but these very faint kind of ground, if you can trust me, these look a little bit like ground glass when you look up close. This was the early set of findings. And after these ground glass appear, this is the same patient on day 5, day 15, and day 20. And this patient, a few days following day 20 CT, did pass away, did die of this disease. But here we see the changes that just continue after 2 weeks, and then after almost 3 weeks, really involving much of the lung with, as I pointed out, very little healthy looking lung left. Carolina mentioned PCRs, and one of the, I think, most important topics that's being discussed this week in the United States is how good are these tests? I would argue, I think I'm not alone in this, that the tests are still not very good. We certainly need more of them, and we've had, I think, a really impressive improvement in our access to testing in many, if not most, communities around the country. It doesn't mean the problem's solved, but I think we have more access to PCR testing, which, as Carolina mentioned, is the testing modality to confirm that a patient has disease at that time. The serologies, those antibodies, are, remember, only really confirming that a patient's body recognizes the virus from the past, but we do PCR testing. And here, I give you the reference. I'll let people look at it if they're interested. But interestingly, this was 205 patients in China. And within those 205 patients, all of whom had a positive test from somewhere, they looked at all of their clinical specimens from a variety of different parts of their body. And they basically said how many of those that were sampled were positive. And I think this is the number that we're seeing being thrown around a lot. It originated from this publication in JAMA back about a month ago. But it showed him, it's very small n, but it showed and was consistent perhaps with sputum of a sensitivity of about perhaps 63 to 72% if we look at nasal swabs and sputum swabs. And the pharyngeal swabs, so just in the back of the throat, really didn't perform well. There's been more interest and more written about how good the tests are in really picking up disease and confirming it, but I think this continues to be a hot topic. So what about duration of illness? Is that why this is a bad disease? Well, what we know with COVID-19 is that these patients really initially have quite mild symptoms, and the mild symptoms do progress over 5 to 7 days. For those with severe disease, we're finding that they're hospitalized after a median of 7 days. And anecdotally, my own experience in our community, has been exactly this, perhaps even a little longer. Patients have some symptoms for the first several days, perhaps even the first week or a bit longer, they do okay. But at some point, they do notice more shortness of breath, perhaps as those ground glass opacities are developing in their chest. And eventually, for a small group of patients, we are seeing really one of the worst complications of lung injury, lung infection, the acute respiratory distress syndrome, or ARDS, which is developing in, again, only a small group of patients, but is the major complication in this disease. And one of the things that we heard about a lot, especially last month, was this idea that this can come on very quickly, meaning a patient can stutter along feeling sick, but stably sick, and then something changes, and suddenly the shortness of breath gets much, much worse, and their oxygen needs suddenly increase. So what about the fact that there's no effective treatment or cure? Is that really what's making SARS-CoV-2 such a bad virus and COVID-19 such a bad disease? So we've heard a lot of discussion with regards to potential treatments, and my job today in just the next 10 minutes is not to give you a full primer in all of the possible treatments, but let me mention 4 or 5 important ones. Remdesivir is, as Carolina mentioned, acting at the viral polymerase, and it's being investigated currently in 6 clinical trials. These are good quality randomized clinical trials, and this is what we need to be able to answer these questions. This is a medication that's being studied in mostly more severe illness, but also some milder to moderate illness in the United States and France and in Asia. And there's some good reason to believe it might work. Some data from both the lab in vitro, but also some animal studies, particularly those published in Nature, related to this particular virus and this particular agent. So I think very promising. I have some experience with remdesivir here locally, without going into the patient details, I can say that this was a patient with severe illness who had a good outcome with this treatment. Carolina mentioned Kaletra. So this is an older HIV combination medication, lopinavir-ritonavir. It's been described in lots of case reports, and it is still actively being evaluated in clinical trials. But I think for many of us, the answer to Kaletra, at least for now, has come from a publication in the middle of March in the New England Journal. This was a good quality, not large, but a moderate-sized study, really a well-done study, randomized control, looking at Kaletra versus those receiving only standard of care, and really failed to show much benefit. Kaletra, I take care of a lot of patients with HIV and have in the past used a lot of Kaletra, but we really don't use Kaletra for HIV anymore if we can avoid it, because it does have some bad GI side effects. And so I think for now, I think most of us are leaning away from using Kaletra, but I think some good quality studies still looking at this, and looking forward to hearing more about this. I think the topic being most actively discussed in the media in the last couple of weeks has, of course, been hydroxychloroquine. As Carolina mentioned, this is an older antimalarial medication. It's still currently used primarily for rheumatologic diseases, so rheumatoid arthritis or systemic lupus. And in what became a blockbuster, essentially observational study, where the researchers in France reported last month an experience where they put most of the patients, or all the patients in their study at their institution, on hydroxychloroquine, and compared that group of patients and their viral clearance, so how quickly they got rid of the virus from their nasal secretions and from their body, and they compared that to patients at a different institution who did not receive hydroxychloroquine. Some also received azithromycin, and there was some potential benefit in how quickly they cleared their virus. This didn't look at hard clinical endpoints, deaths, illness duration, but there have since then been 2 randomized trials reported. A lot of this is pre-publication. And so I'll pause here and I'll quickly mention, as those of us on the clinical side and those of you on the science side are wading through the volumes of information regarding this disease and this virus in the coming weeks, I ask us all to remember that we are living through a really extraordinary time with regards to access to information, where we have the tremendous benefit of being so well connected that when a researcher in China realizes something, I may well know it 24 hours later because they raise their hand and they put it out there. However, those usual checks and balances, including peer review and real scrutiny that we rely on in the world of science and clinical medicine, those really, for the most part, have fallen by the wayside in a rush to get the right information out as fast as possible. And so I point out that the hydroxychloroquine information, I think, is interesting and promising, and there are many good well-designed clinical trials ongoing right now to help answer this question, whether hydroxychloroquine is of benefit. If it is of benefit, in whom is it of benefit? And if there is a group who is benefiting from hydroxychloroquine, who are they? Is it actually as a prevention medicine? Is it during severe disease or a more mild disease? So I think more to come on that. I think certainly worthy of study, but I would say not yet definitively answered. Two other quick treatments to mention. One is tocilizumab, which is a medication also used to dampen the immune system in rheumatologic disorders. It's a very powerful medication. It's a monoclonal antibody. And the idea here is that maybe we can calm down that really intense inflammatory cytokine storm that some patients seem to experience, particularly during their second week of their infection, where they tend to suddenly do worse. And then finally, I think perhaps most exciting in the last couple of weeks has been convalescent plasma. And Carolina mentioned this, but this is the idea of saying that a patient who has recovered from this infection has now antibodies that were formed to help them recover. And those antibodies may actually have use for the next patient as a therapy. And so we actually got guidance last week from the FDA nationally for protocols and ways to basically use this as a therapeutic. And here at Cottage Health, we do now have an expanded access protocol for this, and are looking forward to hopefully using that as a therapeutic in the very, very near future. But at the end of the day, what do I want you to remember? I was a high school teacher before going to medicine, and so I apologize for that up front. But I just want you to remember, if you remember nothing else, what I'm saying today is that at this point, no treatment has yet clearly been shown to be beneficial in high quality clinical trials. I think more is coming, but that is where we are as of today. So what factors impact mortality for a viral infection with no known treatment? Well, let's consider Ebola. So during the outbreak in West Africa, the case fatality rate in West Africa itself was 50 to 90%. Of the 27 patients with Ebola in the United States or Europe, where they received aggressive supportive care, the case fatality rate was much lower, 18%. And again, remember, this is an infection for which there was no specific treatment. And so in the absence of a proven treatment, it's really ICU level care, critical care, supportive care that is going to determine whether a patient does well or unfortunately does not. So moving on to other potential factors that influence COVID-19 being bad, let's think about permanent sequela or disability. We are seeing quite prolonged disease in some of these patients. Those with mild disease are recovering within a couple of weeks. I've had a patient really much better within a week. But most it's taking a couple of weeks. And those who do recover from the most severe disease, of course, implying that unfortunately a very small group are not recovering and are dying from this. But those who do are doing so in 3 to 6 weeks. We've certainly had success stories where patients are intubated. They're on a ventilator, on life support in the ICU for over 2 weeks, sometimes over 3 weeks. And yet they still ultimately are getting better. That's certainly not everybody, but I think this is a prolonged ICU supportive care situation from which some people are doing well. So is it all about infectivity? Is that really what is rendering this such a bad disease? Is it that it's very contagious? Well, those of you who study this, and I realize there are a whole host of experts on this seminar series and on this call probably today, but we think about the R0 is really an estimate of how many people one infected person with a disease is likely to infect themselves. And so I mentioned measles. Measles has a terribly high R0, perhaps up to 20 at times, where one infected patient with measles can infect upwards of 20 other people. We think for COVID-19, the number is somewhere between 2 and 4. And so if we know who has this, and I say that again, if we know who has this, and if we can, put some infection control measures in place, as we learn more about the epidemic, we'll be able to refine this R0. But for right now, we are assuming that the R0 is again in the range of perhaps 2 to 4. Carolina mentioned again some of the viral shedding that we're seeing happening, and this group looked at 9 cases and really studied where in their body the virus was hanging out, where was it being actively replicating, and where were these people PCR positive. The pharynx, the back of the throat, had very high shedding during the first week of symptoms. There was a lot of virus that they sampled from the throat and the lungs, but not from the stool. Blood and urine in these patients never had virus, and there was confirmed to be active replication in the throat. I think most worrisome from a public health perspective is that shedding of the viral RNA outlasted symptoms, meaning even after you were better, they were still finding some of these patients were PCR positive down the road. And I think I just wanted to mention, without going into the study in great detail, but just published last week by the CDC, was one of these, again, super spreader events. And there have been many of these, whether it's the choir in Washington State or these 2 family gatherings in Chicago, Illinois, where, again, those who are interested can review the article, but basically from 2 family gatherings, they had 16 cases and 3 deaths, just remarkable. And so maybe there are some people who end up being super spreaders. This was reported, of course, in New York State, during one of the initial clusters there. But this idea that perhaps not everyone is equally infectious. Is it that it's silently contagious? And so let's talk for a second about the incubation period. Those of you who work with epidemiology or clinical medicine remember this, but the incubation period is the period of time from where a patient develops their from where a patient was exposed to the infection to when they develop their symptoms. And this, I would say, is the best study of this, where these researchers found 101 confirmed cases where they had confidence that they knew when the exposure had happened and when the patients began to have symptoms. And the incubation period, the median was 5.2 days. And they estimated that 97% of those who develop symptoms really do so within the first 10 or 11 days. The famous Diamond Princess, I think, is really important to talk about from an epi perspective, where almost everybody on board was tested, and ultimately 46.5% of those with positive tests had no symptoms when they were tested. So again, to say that again, they tested and they found that about, I think, 19% of everyone on board was positive. But only 46% of those who were positive actually had symptoms. And so, then the question is, how does that break down by age? And again, people can review the slides later, but there was some variability in how old you were and how likely you were to be symptomatic if you had a positive result. Again, this idea that there's potentially some silent transmission. And what about just the idea of poorly understood transmission routes? So from an infection control perspective, I think most know this. But of course, this is a highly contagious virus, and there is real importance in controlling the spread of disease. The CDC recommends that we use standard contact airborne and eye protection, and so we're taking care of these patients in airborne isolation rooms and with N95s, those nice tight masks or PAPRs to keep ourselves and our health environment safe. The WHO is recommending the same. But they're using a simple mask when close to the patient, and really only the airborne precaution when there's going to be aerosols generated. Hand washing and careful disposal of contaminated materials, of course, important in my setting. And just to remind everyone, the alcohol-based disinfectants above 60% concentration do look to be effective, as are some of the commonly used commercial wipes. This study's been talked about a lot from the New England Journal, but here we basically see the viability of SARS-CoV-1, remember the 2002 outbreak, and our current virus, SARS-CoV-2, in a variety of aerosols as well as on a variety of surfaces. The median half-life of the viable virus in the aerosol was about an hour. And no viable virus was detected after 4 hours on copper, 24 hours on cardboard, and 72 hours on plastic and stainless steel. So again, it took up to 3 days for really the virus to look like it was completely gone from plastic and stainless steel. How far does a sneeze travel? This is not particular to SARS-CoV-2, but researchers about 4 years ago in the New England Journal of Medicine looked at how far these puffs of aerosolized materials move around a room after a sneeze, and showed it to be 19 to 26 feet away. So yes, so I know we're all far from one another, and that's probably good for today. Okay, so just finishing up, is COVID-19 a bad disease because of the vulnerability of global or specific populations? This is really what I spend a lot of my professional life, doing, and these are the patients I generally work with. And so, we've seen outbreaks locally as well as around the country, and in fact around the world, where we have a very vulnerable population within an incarcerated persons institution, so within a prison, within a jail. Other populations, those living in resource-limited settings, who have limited access to medical care. I think in the last week around the United States, we're increasingly seeing examples where we have communities that are being hit very hard with this disease, and some of which are, of course, resource-limited settings. And we start to see race being described with some of this discussion as a really important factor. Those who are food and housing insecure, homeless patients, those of advanced age, simply being of advanced age, I think puts you in a vulnerable population. Medical fragility, medical complexity, and of course there are many that I don't mention here. But I would argue this is definitely a part of why COVID-19 is a bad disease, is hitting each of these vulnerable populations and more. The CDC last weekend published some of the geographic differences around the country, our own experience now. This was data as of April 7th. And again, I'm not going to spend time on this slide, but I'd love people to just, if you're interested in this, pull this paper up and look at it. Here, the researchers compared, and they're reporting the number of cases by a variety of jurisdictions the week of March 31st and compared it to April 7th. And then they looked at how much the incidence in each of these jurisdictions changed. Here in California, we had a cumulative incidence of 20.6 per 100,000 2 weeks ago, and then it had jumped to 40 for an uptick of 19. Just to give us a context, New York City itself, the number was up, that cumulative incidence absolute change was 418. Just a remarkable metric of how much their epidemic has just exploded over the time period here. And again, this is a graphic from that study for those who are interested, again, seeing that very steep rate of change in New York City in particular. This is dates versus cumulative incidence. The geographic differences reported in this study, I think, are related to 6 aspects. So, of course, it depends when, and we're going to talk more about this over the seminar series, but when these COVID-19 disease clusters, when the infection is introduced to a community, impacts this. How dense is the population? Of course, that's a big factor in New York City. Age distribution and prevalence of underlying medical conditions within a geographic jurisdiction. What is the community doing to mitigate spread of disease, the ability to even find the disease, the diagnostic testing capacity. And of course, this data from the CDC relies on good reporting practice, where they really do hope to find out about every case. And so case fatality rates, just to start to finish up here. This is data from China, and we've heard this and seen this, but I think the picture really brings it home to us. This is looking at the estimated case fatality rate by decade of life. And I think really this is just as striking, again, as we're all aware, but the vast burden of the mortality in this epidemic is occurring in those over the age of 60, and almost all above those over the age of 50. We've heard the comparison to seasonal flu frequently, but here for context, seasonal flu by age versus the case fatality rates just in the first 6 weeks of this epidemic, strikingly different. And then finally, with regards to case fatality, and I think for those of us with young children, I think mostly reassuring, here you see a breakdown of the Chinese population by decade of life. Here you see where the confirmed cases were in the Chinese experience by decade of life. And here you see the mortality. Again, just striking how much disease there is through middle age, but yet how much death there really was in the older patients. And I think many of you are mathematicians and modelers, and so this, of course, is dear to your heart, but the idea that rates depend on the denominator. As we are testing more and more people, as we find out about asymptomatic or minimally symptomatic patients with disease, we are going to see an impact on our rate of severe disease. And similarly, we're going to continue to see deaths, unfortunately, but our denominator is likely to change substantially over the coming weeks with changes in testing and improved access to testing. And we just need to be ready for that to really analyze that data correctly. And so finally, what about the extent of the pandemic? I'm not going to spend much time on this because experts are going to over the coming weeks, but just for context, a week ago, this was the state of things, 1.2 million around the world, 64,000 deaths. As I prepared for this talk, in almost exactly a week, we'd bumped to 1.8, and of course, much higher already today. In the United States, we jumped from 305,000 to, I think everyone's seen, and even, again, of course, bigger numbers today, but 547,000 around the world. And so just making the point, and we're going to talk in a much more sophisticated way about flattening the curve, but the idea that what we're hoping to do in Santa Barbara, in California, and indeed in parts of the United States not yet severely impacted by this disease is, of course, an attempt to flatten that curve. And so really to summarize, I think I just have 30 seconds, which is I hope I've made a fair argument that for really every one of these measures of severe illness, of badness, I would argue that COVID-19 has proven itself to be a very bad and very severe disease. Thank you very much.
Thank you very much, Lynn. It's been fascinating and frightening having you share this information. We have gone a little over time, and we had hoped to answer a few questions. We need to apologize to Melody, Tracy, and Shatterin that we can't take the questions now. But I have put on the chat window the URL for our website. A number of people have asked, are we going to put recordings of today's webinar online? Now, we will look into that. And if we are able to do so, we will let you know if you sign up for our announcements, and we'll put some information on the website. And so once again, that website address is in the chat window. You're welcome to take a look and copy it down. But for today's session, we need to thank you for coming, to thank our panelists for speaking, the Chancellor for his support, and look forward to welcoming you back next week when we bring another topic in our continuing discussions about the COVID-19 crisis and epidemic. Thanks so much for joining us.
Thank you.
Thank you.
Thank you, Joe. Thank you, everyone.